Okay, we are now going to transition from the air in the water. Our next speaker is Diana Rennison. She is an assistant professor in the Department of Ecology, Behavior, and Evolution at UC San Diego, where she uses genetics and genomic tools in the stickleback, one of the workhorses of evolutionary biology, to trace how selective agents interact to shape the course of evolution. Welcome, Diana.
Thank you. I'm an evolutionary ecologist and a genomicist, so this is not my typical crowd, and I'm really, really grateful to the Cavalli Foundation and the Allen Foundation for the opportunity to have this weekend or a few days to see what the field is doing and all of the great and exciting work that is going on. So today I'll take some time to tell you about the efforts that my lab is undertaking to understand the genetic basis of adaptation to climatic variables, and this is really feeding into this new Allen Discovery Center. All right, so I don't think that I need to convince this crowd that the world is rapidly changing and that environmental conditions are going to often be novel to the organisms that are encountering them. And there's also clear evidence that these shifts in temperature can have direct and indirect effects on the fitness of many organisms. And importantly, many of these effects may be mediated through the nervous system. For example, as water temperature rises due to climate change, a fish may experience changes in neurotransmitter function or brain structure. And this can lead to altered behavior. So due to these changes, a fish may increase its aggression or have impaired predator avoidance behaviors. Yet throughout the history, the evolutionary history of many species, the organisms have encountered a variety of different ecological conditions, particularly when it comes to temperature. So throughout the day, an individual can experience a variety of different temperatures. And we also see variation at the geographic scale or the landscape level. So in the case of a species with a broad distribution, we may have populations that are experiencing and adapting to very distinct temperature regimes. So we really want to understand how organisms adapt to this variation, both on small time scales and across many, many decades or years. So my lab aims to describe the evolutionary response and the underlying architecture. We consider the population dynamics, so things like gene flow. We consider phenotypic plasticity, the spatial distribution of adaptive alleles. But what I'm really going to focus on today is the genetic architecture, so those variants in the genome that explain differences in thermal or other environmental tolerance. So to achieve this, we use the three-spine stickleback, and the stickleback's a really powerful model for evolutionary ecology. So we know that they can rapidly adapt to novel conditions. At the end of the last glacial maxima, marine stickleback colonized newly formed freshwater habitats and rapidly diversified within these habitats. So we know that they can evolve quickly in response to the environment. The stickleback also has great resources. So they have a well-annotated genome. It was Sanger sequenced, so we have chromosomes, we have gene annotation, so we're able to really leverage that to understand the genetic basis. And we also have protocols in place to do CRISPR modifications as well. And lastly, and perhaps most importantly for our purposes here, the stickleback has a really broad geographic range, so that's outlined here. So it's throughout the northern hemisphere, just along the west coast, spanning all the way from Baja California to the northern parts of Alaska. And so, of course, there's really different thermal regimes across that range. So just along the west coast, it's ranging all the way from 3 degrees Celsius to 22 degrees Celsius. So that's an enormous difference in thermal experience. So we're going to really leverage this natural variation to ask about the genetic architecture. So I'm going to walk you through kind of the approach that we take. So first we have to sample stickleback, which involves going outside, throwing traps into the environment, and getting those fish. So we've surveyed fish across multiple climatic gradients. The one that I'm going to talk to you about today is the west coast of North America. So we have 1,840 stickleback that we have sampled across 216 sites spanning from Southern California all the way up to northern British Columbia. And we're in the midst of sampling Alaska, so we will have additional samples that go all the way up the coast. Then we aggregate climatic information. And to do that, we leverage a number of online databases that use satellite-derived data, which includes the WorldClim, TerraClim, and NASA Earth Data databases. And all of these contain variables that describe some aspect of maximum or minimum temperature, seasonality of those variables, or precipitation. And because these variables tend to be highly correlated, we use reduced dimensionality approaches to account for those correlations, so principal component analyses. So then for all of the locations that we have samples, we prepare whole genome resequencing libraries, and then we put that data through our standardized pipeline. So we use an Illumina short read sequencing platform for the sequencing. Once we get those short reads, we align them to the Stickleback reference genome, and then we identify variable sites in the genome. So we ask, is it an A, a C, a G, or a T? Do all of the individuals in our data set have the same nucleotide, or is there variation? And if there's variation, then what does an individual fish have for the genotype at that SNP, the single nucleotide polymorphism in the genome? And after we do all of our quality filtering, we end up with about 5 million SNPs across the Stickleback genome. So once we know what the variation is genetically, we want to ask how that co-varies with temperature or other climatic variables. So we use something called environmental genome-wide association mapping, and the software we use to conduct this analysis is called BayPass. And we use this to identify candidates that are repeatedly associated with environment. And that looks like this in the simplest sense. We're having some sort of variable, for example, temperature, and then we have the genotype at a particular location in the genome. And we're most interested in sites that have a pattern like this, where you have populations that all have a particular genotype that occupy a high temperature environment, and populations at lower temperature have the alternative genotype, and populations that exhibit intermediate temperature habitats have an intermediate genotype. And in this analysis, we control for population structure, so how related different populations are to one another. So we repeat this pattern for every, all 5 million of the SNPs in the genome, and then we can look at it genome-wide. Once we identify candidates, then we can start to interrogate them, and that's where I really leverage my collaboration with the Allen Discovery Center. I'm not a neurobiologist, so leaning on my team to associate this with neurobiological phenotypes. So the data that I'm going to talk to you about today is going to focus on 2 regions in particular. So the Haida Gwaii archipelago, where we have 46 populations and 534 sequenced fish, and California, where we have 29 populations and 365 fish. And there's a couple of reasons for focusing on these 2 regions. So the first of which is we've done population genomic analysis that suggests that they have a distinct evolutionary history. So these are actually independent evolutionary replicates that we can leverage to look for the repeatability of the underlying architecture. And that's because when you do these analyses, there's always the chance of getting a false positive. So if we're going to pursue candidates, we want those really robust ones where we can have evidence from 2 independent replicates. And this is just that population structure analysis. Each row is an individual, and you can see these individuals are from California, and they have this pink and blue ancestry. These are the ones from Haida Gwaii. They have green ancestry, and so you don't see shared probability of assignment to these clusters between the 2 different regions. The other reason for looking at Haida Gwaii and California is they actually have really similar climate gradients. So the absolute values of the climate is very different, but the variation is actually kind of similar. So here is Haida Gwaii. It's an island archipelago that has a mountain chain running down the center of it, and that creates a difference in the climate between the western and the eastern sides of the island. So the western side tends to be colder and wetter, and the eastern side warmer and drier. When we look in California, we see that same or a similar gradient, but in this case, it's north to south. So the southern part of California is much warmer and drier than the northern region of the state. Okay, now jumping into the results. Okay, so many sequences, many dollars results in 2 plots. And on the y-axis, we have the Bayes factor. So this is the strength of the association for a particular marker with our ecological or environmental variables. And we have California and we have Haida Gwaii. These are all of the chromosomes in the Stickleback genome. So there's a couple different colors here. The gray colors are markers in the genome that do not have a significant association with our climatic variables. The orange markers are significant in a single comparison, so Haida Gwaii or California, and then the black ones are candidates that were significantly associated both in California and Haida Gwaii, so there are parallel or are robust, or most robust candidates. So there's a couple things I want you to take home from this plot that has a lot going on. So the first is that there's not a single region of the genome that explains climatic adaptation. It's not the case that we have just one clean peak and that's it. You might see that sometimes when you're mapping a really simple morphological character, but this is a very complex genetic architecture, highly polygenic. So that's the first thing. The second thing is that not all chromosomes are equally important. So for example, this chromosome over here does not have any significant candidates in Haida Gwaii. But repeatedly we see chromosomes 1, chromosomes 4, 7, and 20 in both California and Haida Gwaii having this strong signature and many, many markers having this strong association. And this is really interesting because in stickleback, if you're a stickleback aficionado, these are hot chromosomes because they're repeatedly involved in ecological adaptation, a lot of important trophic traits, anti-defense morphologies, pigmentation, body shape, all of that tends to map often to these chromosomes, so they're kind of really ecologically important. The other thing is that there's certain regions that show a shared signature between California and Haida Gwaii, so we have several of these black candidates that are outliers in both, but then there's certain places that are unique, so you only find them in California or you only find them in Haida Gwaii. So like I said, for the purposes of our initial work, because there's so many candidates in the genome, we wanted to narrow down our list of genes of interest. So we wanted to look at the intersection between California and Haida Gwaii, and we found 142 regions. And within these regions, there were 58 described genes. And by described, I just mean there's strong orthologs in other species so that we can have a particular name and a known function. And this is very hot off the supercomputer, so I haven't looked at all of these candidate regions, and I'm really giving you some just, choice genes that I'm going to tell you very briefly about because we haven't interrogated them at all yet. But since we're interested in the neurobiological leaning functions, I picked a couple of the genes that are these double candidates. So one is GRIA3, which is a glutamate ionotropic receptor. And when we found this gene, I basically went to Google Scholar and started looking at what we know in other species. And this gene's quite interesting because in several other fish species, including mahi-mahi, eels, and trout, it's been linked to variation in migration and other aspects of social learning. So in damselfish, it's involved in cleaning behaviors. So it could be a really interesting candidate to do some behavioral tests to start to look at gene expression in various tissues and do RNA-seq or qPCR. SLC4A7 was another one that popped up, which is involved in the regulation of bicarbonate transport during ocean acidification, and it's been found to be expressed in the olfactory rosette of sea bass, so that was another one that kind of piqued our interest. And AR3A is the circadian regulation of vertebrate cone receptor function gene, so we think it might be involved in the phototransduction cascade, so that's another, kind of interesting gene. But we have a lot of work to do. So the take home so far is really that the architecture is highly polygenic. It's so much of the genome is responding, and that's not surprising because climate is this really amorphous thing that has many indirect and direct effects on organismal function, organismal fitness. And so it would be kind of naive to suggest that there's only one gene that's going to explain the response to such a all-encompassing selective force. But we do find this interesting enrichment on certain chromosomes that are ecologically important. And then we do find the shared signature between Haida Gwaii and California, which I think is very exciting. But it is the case that a lot of that architecture seems to be unique to either of the comparisons, suggesting that there's multiple solutions to similar ecological problems. And yeah, lots to do. So I'm very grateful for my team members in ADC Nice that I'm going to leverage to understand more about what exactly these genes are doing, what's the biological function, and yeah, the behavioral data and the gene expression data I think is really, really going to be exciting in the imaging. And in terms of my lab and our expertise, one thing that we're really excited about is leveraging this to understand how populations are going to respond to future climatic conditions. So looking at something called genomic offset, which is understanding what is the current variation that a population has and what's the allelic frequencies that they need to be able to cope with predicted climatic conditions. So that's the type of modeling that we're really eyeing up to bring this to, management level questions and understanding what is going to happen to this species and related species that occupy similar regions. And the last thing I just wanted to touch on, because we had this prompt, and I feel very ill-equipped to make recommendations to this group of people because it's so outside of my field, but I was so encouraged by the work that people are doing, trying to really mimic natural conditions, starting to look at a variety of different systems, looking at individual level variation, both within a particular site and across various sites. So I think, in 5-10 years, this meeting is just going to be so, so exciting. So thank you to all of you for inspiring me and really getting me excited. And so, of course, I didn't do the work alone. So it was really spearheaded by Caitlin Wise and David Ferranti, who are my 2 PhD students. So they were the ones to do all of the sequencing and the bioinformatic analyses. Of course, we didn't sample that entire coast by ourselves, so we have many people to owe for collecting stickleback samples and helping us with our field work. And then, of course, the funding is what has brought us here and allowed us to undertake this work. So the Paul Allen Frontiers Group, we're super thankful for. I'm really thankful to be a part of this Discovery Center. The California Conservation Genomics Project was what allowed us to first start sampling in California, so I'm thankful to them. And then I've leveraged funding from other projects to make sure we could sample this whole coastline. So, and I would really love to thank you for your attention today.
Any questions for Diana? Yeah, right there.
Hi, so I did really like your talk. You had mentioned that there were alternative genotypes within the fish that were from colder temperatures. Did you notice any phenotypic differences between the fish that you sampled from the warmer temperatures and the colder ones?
So we're still working on the phenotype. So every fish that we have sequenced, we have its body. And so we are in the midst of doing all of our typical morphological measurements and also looking at pigmentation. So stay tuned. We will have those associations for all of that as well.
Thank you.
Any other questions? Yeah, Sean? Ooh, sorry, microphone is back here, yeah.
Okay, great talk. I'm Christine Merlin from Texas A&M University. I was wondering if you did look at other things than just SNPs, such as structural variants, inversions, other really important thing in the genome that could be involved here.
Yes. So we haven't done that yet, but we're in the midst of building our pipelines to look at inversions and copy number variants. So I have a new postdoc, that's his whole thing. So he's starting May 1st, so hopefully we'll have that data soon.
Yeah, so just kind of following up on that, I don't know if you can really tell this, but like, do you see, when you see a SNP variance, do you see the same alleles showing up? And if you've got similar alleles for the ones that match the two, the Northern Island and California, is the polarity the same across the gradient where you can assess that?
Right, yes. So that's also on our to-do list, because yes. So yeah, because it's whole genome data, we can actually say whether the allele itself is the same or if it's the same gene but a different mutation. So we will be able to answer that soon.
I also had a question. You mentioned the temperature as a covariate, but I was wondering what specific measurement of temperature you're using. Is it temperature extremes, averages, winter, summer? and whether in stickleback biology there's any traits that have been established already that would implicate a particular measurement of temperature to tighten that connection between phenotype, environment, and genetics.
Yeah, great question. So what we've done, the world climb, we got all of that. So it gives you the max, the min, and an average temperature, and then there's aspects of seasonality, so a variance component as well. So at this point, what we've done is just fed that all into the PCA, so it's taking it into account because all of those things are correlated to some degree.
Right.
So we can also map them separately and then look to see, and this is something I'm really interested in doing, is maximum temperature giving the same signal as minimum? So is it thermal tolerance in general, or is it hot versus cold tolerance? So that's something that's kind of our next thing. It's all been run, just we haven't picked everything apart yet.
Cool. Are there any other questions? No? Okay, let's thank Diana
Thank
one more
you.
time.